हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी 4th सेमेस्टर बायोकेमिस्ट्री का प्रोटीन कॉन्फ़िगरेशंस का स्टडी करेंगे, जिसका इंट्रोडक्शन है। प्रोटीन आर ए वाइटल माइक्रोमोलिक्यूल प्रेजेंट इन ऑल लिविंग ऑर्गेनिज्म। दे परफॉर्म ए वाइड array of functions essential for life, including catalysis of biochemical reactions, providing structural support, enabling cell signaling and transporting molecules. Protein configurations refers to the three-dimensional arrangement of atoms within a protein molecule. These configurations determine the protein functionality, as the shape of a protein influences how it interacts with other molecules. Understanding protein configurations is critical in biochemistry, as it bridges the gap between molecular biology and cellular processes. This essay explores the concept of protein configurations, its definitions, types, importance, and concludes with significance in biological and medical sciences. Definition: protein configurations is the specific three-dimensional structure a protein assumes after folding into its functional form. This structure is governed by the sequence of amino acids in the polypeptide chain, which determine how the molecule interacts with itself and its environment. Protein can be classified into primary, secondary, tertiary, and quaternary structure based on their level of complexity. Level of protein configurations. First, primary structure. The primary structure is the linear sequence of amino acids linked by peptide bonds. This sequence is determined by the gene encoding by the protein. Variations in this sequence can lead to functional changes or disease, such as sickle cell anemia caused by a single amino acid, substitutions in hemoglobin. Second, secondary structure. The secondary structure refers to the local folding pattern of the polypeptide chain stabilized by hydrogen bond. The two most common types are alpha helix and beta plated sheet. Alpha helix, a coiled structure stabilized by intramolecular hydrogen bonds. Beta plated sheet, a sheet-like structure formed by hydrogen bonds between different segments of the polypeptide chain. Third, tertiary structure. The tertiary structure is overall three-dimensional shape of a protein formed by interaction between the side chains of amino acids. These interactions include hydrophobic interactions, ionic bonds, hydrogen bonds, and disulfide bridges. The tertiary structure is crucial for protein functionality, as it determines the active sites in enzyme or the binding sites in antibodies. First, quaternary structure. Some protein consist of multiple polypeptide chains, each with its own tertiary structure. These chains assemble to form the quaternary structure. Hemoglobin, for example, has 4 subunits that work together to transport oxygen. Factor affecting protein configurations. First, amino acid composition. The sequence and the type of amino acids dictate how the protein folds and its stability. Second, environmental conditions. Temperature, pH, and ionic strength can alter protein configurations. For example, extreme heat can denature protein, causing them to lose their structure and function. Third, chaperones. Molecular chaperones are specialized protein that assist in the proper folding of other proteins, preventing misfolding or aggregations. 4. Mutation. Genetic mutations can lead to changes in the primary sequence, which may result in altered protein folding and function. Importance of protein configurations. First, enzyme activity. The configurations of enzyme determines the shape of their active sites, which is essential for catalyzing specific biochemical reactions. Second, signal transduction. Protein involved in cell signaling rely on their structure to interact with receptor and transmit signals accurately. Third, transport and storage. Protein like hemoglobin and myoglobin require specific configurations to bind and release oxygen efficiently. Fourth, immune response. Antibodies have a specific configurations that allow them to recognize and bind to antigen. Fifth, structural integrity. Fibrous protein like collagen and keratin provide structural support to tissue and cells. Misfolding and disease. Protein misfoldings occurs when a protein fails to attain its correct configurations. This can lead to aggregations and the formation of insoluble deposits, which are associated with various diseases. First, Alzheimer's disease. Second, Parkinson's disease. Third, prion disease. First, Alzheimer's disease. Accumulations of misfolding amyloid beta proteins. Second, Parkinson's disease. Aggregations of alpha synuclein protein. Third, prion disease. Infections misfolding proteins such as in Creutzfeldt-Jakob disease. Understanding misfolding mechanism is crucial for developing therapies for these conditions. Applications of protein configuration studies. First, drug design. Knowledge of protein structure enables the development of drugs that can interact with specific protein, as seen in targeted cancer therapies. Second, biotechnology. Engineered protein are used in industrial processes such as enzymes in detergents or therapeutic protein like insulin. Third, structural biology. Techniques like X-ray, crystallography, and cryo-electron microscopy allows to scientists to determine the protein structure and predict their function. Fourth, genetic engineering. Protein configurations inside aid in designing genetically modified organisms with enhanced traits. Experimental techniques to study protein configurations. First, X-ray crystallography provide high-resolution images of protein structure. Second, nuclear magnetic resonance spectroscopy studies protein in solution to determine their structure and dynamics. Third, cryo-electron microscopy, a powerful tool of visualizing large protein complexes. Fourth, circular dichroism spectroscopy helps in analyze secondary structure of proteins. Conclusion. Protein configurations is a cornerstone of biochemistry, underpinning the functionality of this versatile molecule. The study of protein structure reveals how specific configurations enable proteins to perform diverse roles in living organisms. Misfolding of protein can lead to severe disease, highlighting the importance of proper folding. Advances in technology continue to deepen our understanding of protein configurations, paving the way breakthroughs in medicines, biotechnology, and molecular biology. Proteins are not just building blocks of life, they are dynamic entities that drive biological processes, and their configurations is the key of unlocking their secrets. References: